Thank you, Mark. It's a pleasure to be here. So let me just share my screen. Hello, everyone. My name is Idan. I'm a MSc student in the Hebrew University of Jerusalem, and I would like to present to you my research on the utilization of random peptide mixtures as novel food preservative in cultured meat. Just a second. We are all aware that the environmental damage of the traditional meat industry, and probably most of us believe that the cultured meat is the solution to a sustainable meat agriculture. It is obvious that cultured meat would require less water and land, and with renewable energy solution, the production would be green in terms of green gas emission. But what about the overuse of antibiotics? Many articles argue that cultured meat might reduce antibiotic usage and the emergence of antibiotic resistance bacteria. Yet the question remains as to how we can effectively culture cell tissues for human consumption without relying on antibiotics and without introducing contamination risks. Some argue that to prevent animal source bacteria and residues in the final product, antibiotics should be employed only in the first days of culture. But fighting pathogens only in the early stages is a major risk. Some suggest that just like pharmaceuticals, culture meat should be produced in complete sterile environment. And although it might be possible in small scale, it is not feasible to produce bacteria-free meat in industrial quantities and in reasonable price. Thus, we need to implement safe and resistant-proof antimicrobial agents to the culture meat industry. As Professor Hayuka, my supervisor, has shown in the beginning of this session, our group have developed a new antimicrobial agent called RPMs, random peptide mixtures, that might serve as an alternative to traditional antibiotics. RPMs are very easy and cheap to synthesize, they have broad antimicrobial activity, low resistance development, and it is highly likely that they would be safe for human consumption. In this study, I worked with 4 types of RPMs divided into 2 groups, 20-mer RPMs composed of 20 random amino acids, and a 5-mer lipo RPMs that composed of 5 random amino acids and a palmitic acid tail to increase their hydrophobicity. So what are our demands from culture meat preservative? They must have low toxicity towards the mammalian cells. They must have strong and broad antimicrobial activity. They need to be safe and digestible, and of course they must not induce resistance development. First, we wanted to investigate the effect of RPMs on the mammalian cells viability. The cells I worked with were primary bovine mesenchymal stem cells that are isolated and studied in our collaborator, Dr. Sharon Schlesinger lab. These cells are non-immortalized and very challenging to work with, but we chose them over established cell lines mainly because they have the potential to serve as a versatile model for not only stem cells, but to all kinds of mesenchymal tissues. The first thing I did was to evaluate the RPM's effect on the cell's viability. I conducted a residual viability assay after 24 hours of exposure to the RPMs. And as we can see, the stem cells retain 100% viability at relatively high concentration of different RPMs. To evaluate the interaction of RPMs with the stem cells, I examined them after 24 hours incubation with GFP-labeled mixtures. In this picture taken by confocal microscope, we observed that RPMs penetrate the cells without affecting their morphology. Of course, we were very happy with these results and continue to investigate the RPM's antimicrobial activity in cell culture. To evaluate the RPM's activity, we chose 2 model bacteria, E. coli as gram-negative and Listeria monocytogenes as gram-positive model. We started with MIC, minimum inhibitory concentration assay, and we're very happy to see that RPMs effectively inhibit the growth of both bacteria in concentration that are lower than the maximum concentration tolerated by the stem cells, which was 100 microgram per ml. Next, I chose the most potent mixture of each kind and conducted a bacterial killing assay in the stem cell culture. I inoculated the culture with bacteria and after incubation introduced the RPMs for another 1-hour incubation. Next, the media was serially diluted and plated. While the lipo RPM, the LK5P, was not active in this assay, the 20-mer RPM, the LK20, was very effective. It reduced the bacterial load of E. coli by 3 orders of magnitude and completely eradicated the Listeria. Next, the cell were lysed to evaluate bacteria penetration to the stem cells. As we can see here, even a non-lethal concentration, the bacterial load inside the cell reduced dramatically. It is still not clear whether the RPMs only kills the bacteria before penetration, or rather they also have intercellular activity. Now, intercellular activity is very important because it allows us to fight infections not only in the medium, but inside the mammalian cells. We continue to investigate this and already have some evidence for intercellular activity, so we are very excited about it as well. We intend to use the RPMs as media additive, just like antibiotics is used today in most cell culture. So we wanted to learn more about the RPM's mechanism of action and to understand what happens to the peptides after they kill the bacteria. Do they break down? Do they neutralize? To investigate it, I conducted a series of inoculation and bacterial killing assay in which I repeatedly infected the media with fresh bacteria. I started with the inoculation of cell culture media, and after 1 hour incubation, the tubes were centrifuged. The pellet was serially diluted and plated, while the supernatant was inoculated again, and so on. Our findings show that RPMs maintain their antimicrobial activity along the experiment. We hypothesize that after bacterial membrane disruption, RPMs detach from the membrane fragments and return to the media while keeping their potency. This finding is very important because it shows us that even if our culture is infected, our antimicrobial defense against future infections is not compromised. So we have seen that RPMs could inhibit bacterial growth and eradicate bacteria in concentrations that do not affect the stem cell's viability. Next, we wanted to investigate the digestibility of the mixtures. We picked our most potent RPM, the LK20, and introduced it to pepsin in simulated gastric fluid. After 1 hour incubation, I examined the products in MALDI MS. Here we see the spectrum of LK20 before digestion, and I remind you that we're talking about peptide mixtures, so there are multiple peaks. So just after 1 minute of digestion, we observed a dramatic decrease in the original mixture and a rise in smaller peptides. And after 1 hour, all the LK20 was gone, and we have left with short 5 to 10 amino acid chains that are much less active and would easily break down and absorbed in the small intestine, and we are currently working on proving it. The final thing that I did was to investigate whether resistance can be developed towards the RPMs. For that, I conducted an experimental evolution based on a paper published by Bar Meron, a PhD candidate in our lab. In this experiment, I grew E. coli in sublethal concentrations of RPMs and antibiotics to induce resistant development. Here we see an example of the experimental plate. Each treatment had 3 lines, and each line grew in 4 different concentrations relative to the MIC of each treatment. Every day, the bacteria was diluted to fresh media at the same concentration, and every 4 days, the bacteria that grew in the highest concentration was selected to continue the line and was diluted to fresh media with increased concentration adjusted to their new MIC. After 28 days, the bacteria strains were isolated, and I conducted a new MIC assay. Here we see the MIC fold change of the evolved strains. So resistance towards rifampicin, our positive control, was developed very effectively and rapidly. But what is more interesting is the difference between antibiotics usually used in cell culture and the RPMs. The MIC of the lines that grew with combination of penicillin and streptomycin have increased by 4 to 16 folds, while the LK5P was increased only by 2 folds. The strains that grew with the 20 mer RPM, the LK20, developed no resistance at all. We hypothesized that the huge number of different sequences in the mixture prevent the resistance development. So to conclude, I have shown you today that RPMs penetrate mesenchymal stem cells but did not affect the cell viability nor morphology. RPMs have strong antimicrobial activity in cell culture media, and they are being digested very rapidly in simulated gastric fluid. No resistance was observed against RPMs compared to traditional antibiotics. And overall, RPMs have shown great potential to serve as novel, effective, and safe antimicrobial agent in the culture meat industry. And a little bit about our future plans. We intend to examine the effect of RPMs on the proliferation and differentiation of mesenchymal stem cells, and of course to study them with the differentiated cells. We also want to learn the RPM's effect on the cell gene expression and to continue to investigate their intercellular activity. Another very exciting project is to try to integrate the RPMs into polysaccharide-based microcarriers, and I hope we will have more results to publish soon. The last thing that I want to tell you is that we studied the utilization of RPMs in different platforms, one of which is Prevera, a startup company that aims to use RPMs in all kinds of different food products. They are currently looking for collaborators, so if you're interested, you can contact Gal Shamri. So I would like to thank my supervisor, Professor Tzvi Chayuka, and to all of our lab members, and to you for listening today. Thank you very much.
Thank you very much, Idan, for a wonderful presentation, very well structured and exciting data. There's one question over here. Over there.
Thank you. Nice talk. So you showed that your RPMs penetrate cells, but you perform your experiment just for 48 hours. What about long term?
So as I said earlier, we want to do a long experiment of exposure with the MSCs, but here I have an example of an experiment that I did with different cells called bovine fetal fibroblast cells that are not stem cells but are very similar. And we use a low concentration of RPMs, but as you can see, we have not seen any differences between the treatments and the control in terms of doubling time and viability. And we are expecting very similar results with the mesenchymal stem cells, and we intend to do it in the future.
Okay, so they are not toxic long term.
Currently, where it seems like they are not toxic.
Okay, thank you.
There's a question over there
Thank
from
you very
Toby
much.
that may be an online question, I don't know.
Hello? Oh, something has...
I think that's just a cover.
Yeah,
it still
maybe
works.
we can just leave the cover off. We have indeed an online question by Michelle Hauser. She's asking whether you have tested your RPMs also for activity against mycoplasma.
Well, that's a very good question. I examined them against only one kind of mycoplasma, and they weren't active, sadly. But we attempt to continue to investigate it with different kinds of mycoplasma and different kinds of RPM, and hopefully we will find effective treatment for that as well.
All right. Looking forward to that. Again, Idan, thank you very much for joining online, and great work.
Thank you very much for having me.